I want to first thank the organizer for having me here today to talk to you. And, maybe we took it for granted the conference in presence, but the last 2 years taught us that, Zoom and all this platform killed us. I hate it. And so I really want to thank the organizer, Charles, Elsa, Bernard, for being so brave to put this together, because it's not finished yet, this period, but thanks again. So being the last speakers, there is a good and a bad. Bad that I am the one before the break, and so everybody starts to smell coffee. And the good is that I can go a little bit faster because the introduction is already made. So let me go. So what I'm actually interested in the effect of pollution. And I'm not a physician, I'm a physiologist. And I'm interested in the mechanism that is behind that. So we all know that skin is our largest sensory organ. But unfortunately, as they mentioned, the skin has some issue in the sense that age. And when age, you lose these perceptions. And, all our body age. But the fact that maybe we don't realize that each organ ages at different speed. And there are signs that the skin is actually the one that ages first. Already at the age of 18, you can find some damage in the skin. And so that is, we should protect it earlier that we can. Of course, I'm not going to tell you about skin aging and extrinsic aging. I want just to point out in this slide that while intrinsic aging is mainly genetic, the extrinsic, what I call is, depends on the rest of your life. And if you, say the rest, of course, we have to introduce now the exposome skin aging concept. And you can see in the slide, I will try not to give you the back. And so let's see if we can do without, I cannot do the pointer like this, but anyways, so you can see in the bottom that is the concept is a very nice graphical design, where among them, there is for sure a pollution part of that. So pollution and skin has been a topic for the last few decades, but how important was in the literature. If you look at PubMed, this is just a screenshot that I did, I think, 10 days ago, when Charles was really assaulting me with all the emails that I had to send the slides on time. And there are over 7,000 papers published. But if you go through the slides, you see that in the 80s, really nobody cared too much about skin and pollution, because it doesn't do anything. Mainly was related pollution to lung. But of course, when they start to increase this interest, around 100 papers were published in 2000. But if you look at last year, 512 papers were published related to skin and pollution. Why this? It's not because we become all green all of a sudden. It's just because we start to see the damages. And since we start to see the damages, now we are interested to see if we can prevent this or at least stop it. So that is, pollution is one word, but means a lot. That is the main pollutants grouped by the USDA, but EPA. But I add also UV because, as we all know, it's not maybe a pollutant, but it's for sure stressors. Maybe in the next Skinalliance, we will talk also about a new pollutant, that is the microplastics, because now we know that we are exposed to microplastic, and there are start to have data that you find microplastic in the blood. And as Lorraine mentioned, when something is systemic, it can reach the skin from the inside out, okay? The important thing is that maybe one of the take-home messages, each pollutant acts differently. So there are some that can penetrate, some that cannot penetrate the skin. And in general, this slide is a generic effect of pollutant to the skin. Here are all the pollutants. In general, we have 4 pathways that are activated. We know that there is barrier perturbation, there is increase of oxidative stress, inflammation, and also the age receptor activation. But if you summarize even more, you can have two pathways that they go together. One is inflammation, one is oxidative stress. And actually, if these two pathways you put together, you want to summarize even more, we came out with this oxinflammation pathway that is, again, like it happened in science many times, a chicken-egg puzzle, because we don't really know who comes first, who comes second, but we know that they feed each other in this mechanism. So said that, the important thing that maybe we don't realize is that the skin is a very strong organ, as Christos also mentioned. Is very strong in terms also of oxidative protection. We have a fantastic enzymatic antioxidant system that is summarized on the left side, at least in my left side. And also you have a non-enzymatic system. One thing that we should mention is, first, all these enzymatic enzymes that protect the skin are actually regulated mainly by one transcription factor. And please remember this, because I will come back to this later, NRF2, okay? But the other thing that is important to mention is that all the non-enzymatic and enzymatic, they are not really unfriendly. They talk to each other. That's why it's important that when you think of some supplements, whatever you want to take systemically or topically, one ingredient is not enough. Because we all know that, for example, vitamin C is very important to recycle vitamin E. We know that ubiquinol is important also for the recycling of vitamin E. We know that the thiol cycle helps vitamin C formation. So they talk to each other. That is important that you mentioned. And of course, when they are used too much, because we are exposed to too much stress, and skin is weak. And that was a nice Nature paper where they highlights how pollution can really affect the skin through depleting the defense that you have in the skin. So said that, the main pollutant that we are interested is particulate matter and ozone. I wanted to show you how we think is one of the mechanisms how they act to the skin. So the fact that particulate matter can enter the skin is still a big debate. And so I'm not going to take part, never do that, especially in public. But what I want to show you the both sides. We put together this review where we mainly said that maybe particulate matter are not able to penetrate the skin unless it is disrupted. And of course, once disrupted, you start all that pathway that I mentioned to you. But there are also evidences that show that maybe particulate matter can go through the hair follicle. We have to think that anyway is a very small percentage of area that is covered by the hair follicle compared to the whole skin. Or others have shown in this JCI inside paper that maybe the particle they don't go inside, but what is attached to the particle maybe can go through the skin. So independently of they go through or they don't go through, they have an effect. In fact, it has been shown that if you live in an urban area that is a Krugman group, you show that you have an increase of facial wrinkling. This was not only paper, there was another Chinese paper that showed that depending on if you live in a high polluted or low polluted, you have an increase of damage in as measured with, don't know, it's a little bit out of focus maybe, but is the cheeks and also the keratosis in the hand and several markers. And then we, in vitro study, we showed that also in this case, using 3D models, the particulates could induce that are markers mainly of inflammation. So it really have an effect. Actually, we even, so what I want, so if they go in, either from the inside out or from the outside in, we want to understand what happened when they are in contact with a cell. So these are a study that we did with keratinocytes, and if you give the particles to them, actually it was striking to see all these particles that are the arrows showing to you, that they were really inside the cell. So what happened when they are inside the cell? They actually induce, as you can see here, pro-inflammatory cytokines by IL-1 alpha in a dose and time-dependent manner. But what was striking is we found that there was an increase also of peroxidation. And remember, peroxidation, so all these pollutants, they have to have a second messenger to bring the damage to the cell. So peroxidation is for sure one of them, and I will show you later also why. But look, if we use in the bottom image, DFO, for example, just to chelate iron, we found that there is not as much peroxidation. So most likely the iron that is attached to the particles is one of the driving force of the toxicity. But as I mentioned to you, the skin is strong, so it should defend itself. So if you go to measure the transcription factor that I told you, NRF2, if you go, why we want to measure how much this transcription factor binds to the DNA so then can transcribe for our defense, we found that actually the more particulate you give, the less is attached to the DNA. And that was very striking. I said, why? Why you don't defend it yourself? And so, NRF2 is a master defense cellular regulator. So what we found, and that is just to make this first part short, the story of NRF2 is nice in the A plot. Is nice sitting in the cytoplasm with his friend Kip, and they hug each other. And then if they don't use, it gets degraded. Then in the B part, if you have a stress, actually say bye to Kip, it translocate to the nucleus, and there it binds to the DNA where it transcribed for our defense mechanism. So what we found that, sorry for the back, we found that actually NRF2 is affected by the peroxidation that binds to the NRF2. And then when it translocates, this change of conformation will not let bind to the DNA. So even if it translocates, it's not functional. So that was one way that we could explain why there was this not defensive effect. The other pollutant that we are interested in is ozone. Completely different mechanism. When ozone hits the skin, it will never penetrate through the skin. But what it does, and promise I'm not going through the chemistry, what it does, it produces mainly two mediators that are two aldehydes and hydrogen peroxide. We all know that hydrogen peroxide is a ROS, is an oxidative stress marker, and also the aldehydes can be very reactive. And actually, it's true that it's not just a dogma. We measure in skin that there was, after ozone exposure, there was an increase of hydrogen peroxide and also of 4-hydroxynonenal. 4-hydroxynonenal is an alpha-beta unsaturated aldehyde, super reactive. In fact, why I want to underline this? Because this aldehyde is we believe, one, as I report here, one of the main mediators of all pollutants, toxicity. What it does, it can bind to histidine lysine proteins and it changes the conformation of the protein. And your biology class, for sure, you remember that when you change a protein, you have two ways to do. Either you get rid of the protein through the proteasome and you degrade it, or the protein will stay there, but since it's changed, will change function, so will not work as it should. And that is a damage that you carry on in your cell. And indeed, there is this study, that retrospective study, that what they show that there was an increase of ER visit for skin condition in this China for 7,000 patients, while the pollution was going up. And actually, the pollutant that was more influencing the visit to the ER for skin condition and was here are the skin condition was ozone. And after that paper, also myself with colleagues in Canada, we confirmed this with a much smaller population. And then there is a most recent study from Jean Kreutzmann where I showed that there was an increase of wrinkle formation if you live in area with a high ozone effect. But these are all retrospective studies. There was not really a direct study on ozone on skin. And so we did it. So we want to do this study to see, one, if there is really this effect, and if there wasn't, I had to change field. And then, if you can prevent. So we did this study with some antioxidant mixture. As I mentioned to you, we believe in the mixture. And the human subjects were exposed to ozone on the arms, because, as you know, it's also toxic to breathing. And we measured mainly the markers that I mentioned to you earlier, but also others. So you can go to the is a 2017 GID paper. So we show that is in the plot there is the average of 15 subjects, and instead there are just 4 more representative histology, immunofluorescence histology data. You can see that with ozone it lights up, that is 4-AGNE, that's a very toxic aldehyde, and you are able to prevent if you use topical application. We confirm that also for NF-κB, that if you don't know, this guy is the master regulator for pro-inflammatory pathways in our cells. And again, we have more or less the same pattern. The same we had for MMP, the active form of MMP9, that it degraded collagen and then it can lead to wrinkle formation. But unfortunately, that was one pollutant. I mean, we cannot go out and say, what is the pollutant of the day, and today I have to protect against that. No, we are exposed to all pollutants. So since we are exposed to all pollutants, they act differently. So you need to understand if they have an additive effect, for example. And so we did, and to confirm that, we did this microRNOmic study where we took a skin explant exposed to UV or diesel exhaust or ozone, and we checked the pattern. You don't need to be an expert in this, just look at the color and where they, to understand that they have different patterns based on the pollutant that you use. And that is the first study ever done on microRNA. So microRNA are very important mediator of cells, how they talk. So this is just published this year. But so then we decided, okay, let's do our pollutome, we call the pollutome study, where in our lab we have a lot of instruments, a lot of toys, as I say. So we decided to add to the UV light also diesel engine and ozone, and see if there is really an additive effect. That was done in skin explants. We have the doses, but there is a reference, and I will be happy to give you more info if you are interested in the paper and send it to you. And then, of course, again, let's see if you can prevent with an antioxidant mixture. So that was just the picture that my students sent it to me to show me how happy they were about this experiment, because it was very busy. And then we measure more or less the same marker that I showed you earlier. Just to cut the time, I go just on the quantification. These are a lot of data. You can see in the bottom, if you look at the last one, where there are all the pollutants, you can see that there is really an additive effect, and that is explained because all the pollutants act differently. And as you know, if they have different pathways, then they will bring to a sum, additive effect. Instead, if they will go all to the same, like a funnel effect, then you will have only one effect and not an additive. So we show that as, again, our super good marker and very noxious aldehyde for hydroxynonenal increased. And then we showed this for NF-κB. And we showed also, as mentioned earlier, is very important filaggrin. And you can see how nice is the red color of the filaggrin that you loses with the pollutants, and you are able to prevent or replenish with the mix. And then...
Don't worry, I'm not Will Smith. I'm not going to do it. It's not a Will
It's
Smith
the last
moment.
one. It's the last one. Yeah, but you scared me a little bit, because it's kind of popular now to do that. So, but no jokes, promise. So I actually want to say that the main part of where we believe it happens is that NRF2 and NF-κB, they talk to each other. And if you are able to increase one, you prevent the damage that we have seen. So we believe in NRF2 as a possible mechanism target. And, but that is the last and most important slide, is who did the job. I didn't do anything. I just presented it. And I have a fantastic group, but they are sometimes in Ferrara, sometimes in North Carolina, sometimes talk in the airport. But I am very lucky to have them, and I thank them to do this work. Thank you very much.